Hello friend, this is Dr. Pankaj Kumar, your mentor for today's session. In this session, we will be talking about fertilization, in which we will be talking about how male gamete is going to fuse with the female gamete, resulting into the formation of egg and the endosperm, right? So first of all, let's learn the sequence of events. So the very first thing, the pollination is going to happen, right? And as a result of pollination, the pollen grains is going to germinate on stigma. Now, what are the conditions which is essential for the germination of pollen grains on a stigma? Number one, it should be relative humidity. Number two, there must be a carbohydrate, calcium and boron, because carbohydrate, calcium and boron is required for the growth of the pollen tube. Okay, so once it germinates, what will happen? In the previous section also we have seen in the case of microsporogenesis that the pectocellulose will grow and they will form as a pollen tube, and once the pollen tube is going to form, within the pollen tube the generative cell and the vegetative cell will go, and eventually the two male gametes are going to be formed, that we know, right? Now see, this pollen tube will release the male gamete at the micropyle. Now, on the very basis of mode of entry of pollen tube into ovule, three types has been recognized. What are they? First of all, porogamy. Now, what is porogamy? In case of porogamy, what happens that the pollen tube is going to enter directly through the micropyle, and that is the most common one. Okay. Then second is what we call chalezogamy. In case of chalezogamy, what happens that the initial entry of pollen tube is going to occur through chalaza, as you can see in the diagram. So it is entering through chalaza, right? But mind it is not entering directly, rather it is coming across the embryo sac, and eventually the entry is going to occur through the micropyle only. But on the basis of initial entry, we call them it is a chalezogamy, and the typical example is casuarina. Okay. Then we have a mesogamy. In case of mesogamy, what happens? That the entry is going to occur through the integuments, as again you can see in the diagram. But once again the same thing. The piercing is going to happen through the integuments, but eventually it transfer across the embryo sac and enter through the micropyle only, right? And the typical example what we call cucurbits, where we find the mesogamy. Okay. Now let's learn two terms. One what we call obturator. Now what is obturator? What happens when the pollen tube is growing, then there are some ovular tissue which directs the pollen tube towards the micropyle. So that tissue is actually called as a obturator. And there is a one term what we call siphonogamy. So whenever the pollen or whenever the male gametes are actually being transferred with the help of some pollen tube, right, because we know very well that male gametes are not motile. We need some structure to take it to the female gamete. So whenever that structure is through the pollen tube, that particular entire thing is actually called as a siphonogamy, right? Now let's talk about the fertilization. We know very well that the fusion of male and female gamete is actually called fertilization, and first of all it was observed by Strasburger, right? Now let's try to understand that what are the sequence of events that is going to happen during the fertilization. As we have seen that pollen tube is going to release the two male gamete, and the structure that is going to receive the two male gamete is a synergid cells. Mind it, entry of pollen tube is always chemotactic. That means it is the male gametes are going to be attracted by certain chemicals. In most of the cases it is a malic acid, right? For instance, if you see in case of bryophyte it is a sucrose. But everywhere other bryophyte, if you talk about pteridophyte, gymnosperm, angiosperm, in case it is the malic acid that is going to attract the male gamete. Okay. So as you can see in the diagram as well, so these are released and one of the two synergid cells is going to receive both these male gametes. Okay. So out of these, one male gamete will fuse to the egg and it will form zygote, that is the diploid structure. And since there is a fusion, so this particular event what we call syngamy, or we also call them generative fertilization. And as I told you earlier, it was discovered by Strasburger. So this was the fate of the first nuclei, or one of the nuclei, one of the two nuclei that is released from the pollen tube. Okay, now what happens that the second male gamete fuses with the secondary nucleus. So secondary nucleus is already a dikaryotic structure, N plus N structure. Okay, so when they fuse, so the three nuclei is going to fuse together. So that is why this particular event is actually called as a triple fusion. Okay, and this triple fusion is also called as a vegetative fertilization. So earlier it was a generative one, but it is a vegetative fertilization. And as a result of fusion, the very structure which is formed, what we call primary endosperm cell, that eventually leads to the formation of the endosperm, which we will discuss in a subsequent lesson. So it's very clear that here the fusion is going to occur two times. One, the fusion between egg and the male gamete, and second, the fusion between the polar nuclei and the male gamete. Okay, since the fusion is going to occur two times, this entire event is actually called as a double fertilization, right? And it was observed by Nowotny, right? So this entire event is actually, we call them the double fertilization, right? So what we can say, that male gamete and egg, this will form zygote, right? And this entire event is actually called as a syngamy. Similarly, male gamete plus secondary nucleus, this is going to form primary endosperm cell, and the event is actually called as a triple fusion. Okay, so this is all about the fertilization. Now, what are the significance of fertilization? You see, fertilization or sexual reproduction, actually they result in the formation of recombination or new combination or new variants is going to produce, which is very, very important for the very process of organic evolution. So that's why it is very, very important. It also helps in the maintenance of chromosome number, because you see, male gamete is coming with a half chromosome, female gamete is coming with the half chromosome. So the very maintenance of the chromosome number across the species, the onus lies on the very process of fertilization only. Okay, and we know very well that after fertilization the fruits are formed. So that is also one of the significance, right? Now let's have a look some of the post fertilization changes. Okay, and in NEET the questions are most of the time are asked from this particular arena. So you have to keep these facts in your mind. For instance, ovule will get converted into seed after fertilization. Similarly, egg cell will get converted into embryo, right? Secondary nucleus will get converted into endosperm. Mind it, endosperm is a triploid tissue. Ovule will get converted into fruit. So these are the important changes. But there are some minor changes as well. For instance, the outer integuments will develop into outer seed coat. It is also known as testa. The inner integument is going to develop into tegmen or inner seed coat, right? So there are certain parts which shows no changes. For instance, take an example of funicle, hilum, micropyle. You can find all these three things within the ovule. So there is no changes in that, right? And there are certain structures which disorganize after the fertilization. For instance, the moment fertilization happen, synergy cell don't have any role, so they disorganize. Similarly, the antipodal cell also disorganize because they also do not have any role. Okay, nucellus, that is the insider tissue within the ovule, that also disorganize. But there are some cases in which what happens, nucella persist. Okay, and when the nucella persist, we use the term what we call perisperm, and that is found in Nymphaceae, right? Same family in which the lotus is there, right? So this is what we call perisperm. Okay, you may be wondering that if these cells were have to disorganize, then why they are there? Actually, they are there just to meet the eventuality. The role of synergid cell is very much clear because they receive the male gamete and they actually helps in transfer the male gamete to the egg and thereby helping in the process of fertilization. But what about the antipodal cell? Suppose for any reason the egg cell is actually get destroyed, okay, or egg apparatus itself is going to be destroyed. Then the plants or embryo sac has some alternative methods in which they offer the antipodal cells. The antipodal cells can also combine with the male gamete and thereby forming an embryo. We will talk about all these things in very detail in a special mode of reproductions, right? That is in the subsequent chapter. So that's all. I hope the very process of fertilization is absolutely clear at your end, right? Two things once again you have to keep in your mind the very phenomena of double fertilization and triple fusion. That's all for today. Don't forget to like and subscribe this channel, right? Thank you.